Hello, everyone. Thanks very much for coming. So I'm going to talk to you today about Rusty Genomics. So that's Rust in the Biosciences. And I'm Lisa Crossman, and I work on the Norwich Research Park. So in Norwich, we have a pub for every day of the year and a church for every Sunday. We also have a castle, some crabs, and we have Josh Sargent, who's your World Cup football soccer player, and we have the headquarters of Avengers from the Age of Ultron, and Netflix's Jingle Jangle was filmed on one of our oldest streets. So we're located in the east of England, if you ever want to come by. But today I'm going to talk to you about DNA sequencing. And DNA sequencing is important because the structure of the DNA was solved by Watson and Crick and Rosalind Franklin back in the 1950s. And DNA itself is made up of chemical building blocks of three types. So there's a phosphate group, a sugar group, and one of four nitrogen bases. And the sequence itself is a string of just A, T, C, and G. And these are the bases on the sequence. And DNA structure itself is a double helix where you have the nitrogen bases pairing with each other. So A always goes with T and C always with G. And all of the DNA sequence in an organism is its genome or genome on this side of the pond. So these days, we have lots and lots of genomes. And the genomic information has got larger and larger. So it constitutes really big data. And we have various types of data, which are from different organisms. And we study them in different ways, depending on what we want to find out. So, of course, human genome data is really big, and we study it to increase our understanding of life and also diseases. We also have plant genome data, which is usually studied for agricultural reasons. And we have microbiome data, which can be really large, much of which is human gut microbiome data, because we want to know about good bacteria in the gut. And we also study a lot of separate species for things like disease tracking. For example, 2 different species of one bacterium have approximately the same genetic distance between us and a sea sponge. So it's quite interesting. And the size of the data has really seen an explosive increase lately, so that 5 years ago, about 35 gigs of data was a lot, and now we're talking about 3 terabytes of data being quite a lot. And a single tumor cancer sequence can be 2 terabytes of data. So I'm originally a microbiologist studying bacteria, viruses, and fungi. And at one point I went to an art exhibition where the art was made out of book pages. And I was really struck by this exhibit, which potentially sums up why I'm interested in organisms that you can't see. So the smallest creatures cause the greatest concern. And nowhere has it been more true than for COVID. And COVID's been good for some things because it's really ramped up the possibility of sequencing capability across the world for disease surveillance. And you can see here with the light green that there's really high availability across the world. And 77% of countries can now sequence the COVID virus, SARS-CoV-2, in country. And 22% have access to timely sequencing with international referrals. And only 1% of countries have no capability or timely access. And they tend to be in war-torn regions. So DNA sequencing, the method, was originally devised by Fred Sanger. And here's Fred outside the Wellcome Trust Sanger Institute in Cambridge, UK, where I used to work. And Fred won 2 Nobel Prizes for his work in sequencing methods, and he published them in the 1970s. But nearly all sequencing is not done using his method anymore. Although it's still accepted as the gold standard, it's vastly more expensive. So how is sequencing done? So the most popular technology platform for sequencing is the Illumina short read sequencing from the company Illumina. and they say they own about 90% market share. There's also PacBio by Pacific Biosciences, which is long-read sequencing, although a short-read version came out earlier this year, and we've still to evaluate data on that at the moment. There's also Oxford Nanopore, which provides much longer sequencing reads, and we'll shortly come back to the long reads. So this is how the short-read Illumina sequencing works. Illumina sequencing is also known as the sequencing by synthesis, whereby your piece of DNA that you want to sequence is chopped up into very small pieces and then immobilized on a glass slide. And then in a method which sort of hijacks nature, as we can see, the pieces we attach to the glass slide become like a template we can copy. And each base is labeled with its own colored fluorescent dye marker. So we actually sequence the attached DNA by adding one base at a time of the complementary base, so A pairing with T and C with G, and so on. As the base becomes attached, the dye is then chopped off and excited by a laser to produce images. And as we go along processively, each base at a time, we can see the actual sequence. And each of these produced sequences are known as reads. And this takes place on the glass slide of millions and millions of sequences at the same time, which is then read by computer to create the actual sequence reads. So the Oxford Nanopore long-read sequencing works in a completely different way. We have this small and really cool gadget, which just plugs into your laptop so you can sequence anywhere. And the size of it is about sort of that big. And what happens during the sequence is that the actual DNA to be sequenced goes through a nanopore in a membrane, and that causes a current change, which can be measured by a semiconductor in the base of the device. So here's a pair of Oxford Nanopore MinIONs, and they also produce other devices, such as the one with the screen on it, and they're bringing out some new ones, which are really small, that you can attach to a phone or an iPad. And the idea is that you could actually carry out DNA sequencing away from the lab, so maybe even in the clinic or in a hospital. So you might want to know what the data actually looks like. And the short read data from Illumina looks like this on the top. What you can see is the actual sequence here with the A, T, Gs, and Cs. And you get around 100 to 150 base pairs of the short read, depending on the machine model. Underneath, you also get a quality score, which the best quality is towards the beginning of the alphabet. And as you get towards the end of the alphabet, those are the worst qualities. So you can see that the quality is dropping towards the end of the sequence read. And in fact, that always happens. So underneath, you can also see the long read data on the squiggle plot, as it's known. And this is ascribed to the actual sequence by being called by machine learning as groups of 5 bases, or 5 mers originally, but now, in fact, the call is on 6 mers. And the reads are able to be much longer, and that benefits us later when we're doing our bioinformatics. However, the accuracy of the sequence has traditionally been quite a bit lower than for Illumina data, but it's reportedly much higher in newer data. And the reads can be up to about 4 megabases, 4 million bases. And in fact, there's a healthy amount of competition of who can get the longest read. But it does become difficult to prepare good DNA to go into the sequencing process, because very long molecules of DNA are subject to breaking mechanically into smaller pieces during preparation. So like moving them about with pipettes and so on can break them. So one of the very big areas in my biology currently is disease surveillance. And we're very well aware of that lately because of identifying variants in the DNA sequence. And I should quickly point out that although DNA is the genetic material in nearly all organisms, RNA is the genetic material in some viruses, which includes the COVID virus, SARS-CoV-2. So in order to sequence the COVID-2, we first have to convert the RNA to DNA. But once you have your DNA, then the DNA translates into protein. So on the top, we can see the DNA sequence, and each set of 3 bases specifies a single amino acid, which is part of the protein. Here, as you can see, if one of the bases changes, which this can happen in nature, a bit like when you're copying files from one location to another, an error occurs during copying. And so the DNA sequence can change, but not always gives a completely different amino acid. And in this picture, we can see that the change has specified for us a different amino acid. And that is the variant. So what does it mean? On the left, we can see a picture of SARS-CoV-2, which we are often shown in the UK on the television. And the red blobs on the outside are actually a spike protein, which is a protein which attaches to our receptors on our cells. So what do the variants that we're talking about actually do in terms of the protein? Well, you can see in work by Cog UK here that some different amino acids are shown on the structure. And the differences here are the differences between the original spike protein and the Omicron spike protein. And what happens is that sometimes the amino acids changes makes no difference. Sometimes the amino acid that has changed is a different type of amino acid, so it might be bulkier or it might have a charge on it, and that can cause the structure of the protein to bend or change in some way. So there's a very slight possibility that the protein could structurally twist or something due to a variant which might affect our immune system ability to recognize it. So another thing we're often doing is part of epidemiology, which is the study of disease in communities. And you can understand this best if you think about the World Health Organization variants and also about family trees. And I'm sure you can all remember the alpha, beta, gamma, and the delta versions of COVID viruses. And those are shown in the kind of blue of this family tree towards the bottom. And what we can see is that since about December 2021, everything has all been Omicron. And you can also see that although they are all Omicron, there's some sections which are sort of distinct, and they've been given kind of nicknames which aren't really WHO nicknames. And the latest variant that's out there has been called Pyrrhula or Pyrrhula, and you can see that this has actually been hiding out somewhere because it's come from a branch that was further back in time. But just before I left the UK on Friday, I heard that 82% of all the UK cases of Pyrrhula were found in a single care home, and also that luckily nobody from that care home was hospitalized or died. So it's not all about variants, especially in new sequences and in places where you want to find out functional information. And in those cases, you usually start with an assembly. So in the assembly, we merge all the reads together to create one long sequence so we can study it further. And this is somewhat like a jigsaw puzzle. So we start with k-mers. So if you imagine the whole of our stretch of DNA, we split this up into small, tiny pieces of DNA, and we're describing them as a sliding window with a sliding window of 1. And you can see here what that means. So also, if you imagined it being a word, such as happiness, we can create our assembly with a graph theory in what is known as a De Bruijn graph, and this actually recreates for us the final sequence. However, it's actually a very big compute job, and in particular, that takes up an awful lot of memory. And there's also a big problem with Illumina short read sequence with repeats, and the repeats are actually common in nature. So that when you come with a graph, there are alternative paths to follow because of the repeats, and the assembler has to just cut the graph and give us 2 smaller pieces. So we don't know the order of those pieces in the final sequence, which isn't terribly helpful if you have a very large amount of small cut pieces. So always looking for really great assembly programs that are not so resource intensive. So after we've done the assembly, we can carry out gene prediction. And the DNA itself is read in groups of 3 bases, if you remember from variants we were talking about before. And this is known as frames, where we have 3 forward frames and 3 reverse frames on the sequence, where the first frame starts at base 0, the second at base 1, and the third at base 2, and the reverse frames are read backwards. So this is the subject of a Rust Foundation project grant, which I was on. And here you have the sequence of a phenylaria bacterium, which I recently published on with a collaborating group, and it's actually been found to be quite important in prostate cancer. Underneath you can see where the genes have been predicted on the sequence, and knowing where the genes are gives us a good clue as to what the organism's actually doing. So Margaret Dayhoff was an American scientist who's often described as the first bioinformatician, and she put forward a single letter code for each amino acid in a protein. And this enabled computers at the time, which worked on punch cards, to store the sequence on the punch cards. Luckily, we don't have to do that anymore, but bioinformatics as a field didn't really take off until we had suitable amounts of data, which came when the cost of sequencing really dropped in the late 2000s. So bioinformatics is not really for you if you like to learn something and become an expert in it, because you're always going to need to keep learning. It's really innovative. And even if the developers aren't innovating constantly, then the sequencing companies are always innovating and chucking new things at you, providing you with different data in different ways and amounts. One point here for Rust is that I think I read somewhere that Rust is changing too fast for some industries to adopt, but it's not very much of a problem in bioinformatics because everything else is all moving on really fast in any case. So who or what is a typical bioinformatician? Well, I'd say it's probably not me, but the field had to be formed quite quickly from 2 other fields of biology and computer science, which kind of smashed together in the middle to provide us with the bioinformaticians. So there are people coming through now who have actually got bioinformatics degrees, but the content of those degrees tend to be quite different depending on where they went. So at one end, we have the biologists. Many of them are still in the lab. They want something really easy to run. They don't really have time to keep up with the field, and they need quite a bit of direction. Then we have the whole spectrum through to people who are mainly writing programs. And there's always a bit of a trade-off between speed and quality in bioinformatics, but the biologists are always wanting speed and quality, which is not so easy. So I got into Rust, really, when I was asked to carry out a process which wanted to be really quick, and I needed to count some of those k-mers that we were talking about earlier. So originally I thought, I'm not going to be able to do this, because my bioinformatics was Python and R, and a bit of very old Perl. So I thought, I'm going to have to learn something else. And that was when I started looking around and found Rust. And there's some things about Rust that really appeal to bioinformaticians. So we like bioinformatics libraries to carry out our very common tasks that we have to do. And there is Rust Bio. We really like cargo because installing packages is a horrible bioinformatics problem. We aren't really IT specialists, so installing is relatively difficult anyway, and quite a lot of people don't even have admin rights. Most of our tools have a lot of dependencies, which each have a lot of versions, and the version can be really important. Then we have other tools which use the same packages in different versions. So some of the versions can be really finely balanced. You could spend a long time installing something and then discover it's not useful anyway. And therefore, we use a lot of Anaconda container environments to keep different versions of everything in separate containers. And I have about 60 containers, including 2 Rust containers. And I find that Rust works really well with Conda, and you can also keep different Rust versions as well, so that's brilliant. Another thing that really appeals is the way you can integrate other programming languages, especially Python, which is really popular now in bioinformatics. And some people, I think, would really like inline Python, and they're very fond of Jupyter notebooks. And integrating R is also important because we really like it for doing graphs and stats. But one of the major things which is really appealing about Rust is its speed. For example, in the computer language benchmarks game, 4 of those related directly to bioinformatics. They are generating and writing random sequences, writing their reverse complement, and hash table update and k-nucleotide strings. And when I was looking at this table, I was really struck by how well Rust is performing compared to Python and, of course, Perl. So one of the most common things that we have to do is changing file formats. And here is one format on the left, which is known as the EMBL format. And this is a very precisely described type of file, which is kept in the sequence databases in GenBank in Maryland and in EMBL in Heidelberg and in Japan. And what we're trying to do here is change the format on the left to the format on the right. And you can see that the EMBL format holds information about genes and where they are on the sequence. And the other type of format, the DNA FASTA format, only contains the DNA sequence as one long string without any numbering or spaces. So I did some benchmarking on some scripts that I wrote in Rust and in BioPython. And I was really excited to find that the speed for the Rust was much quicker, although it had quite a lot of lines, but I haven't really tried to get that down at all, and there's some comments and so on. And I've been learning Python for a lot longer, so you'd possibly expect the Python script to be better. But then I looked at it again and thought, well, does it really make that much difference? Because I'm going to have to type into the command line anyway, and, sort of half a blink of an eye or a blink of an eye, is it that much? But then I discovered definitely yes, because at one point I was converting 10,000 separate bacterial genome files of EMBL format to FASTA format. And if you did that on a single thread, in Rust it would take 25 minutes, but in Python it would take 4 hours. So obviously you wouldn't do that on a single thread, one after another, but clearly we're using much more resource. So there are some available popular bioinformatics crates, but obviously there's a lot less than for Python. So you have Rust Bio, but there's also Needletail, which I really like, it's really fast. And I'm quite excited about FastQ streaming tools for the reads. And there's one really popular tool, SourMash, which can identify bacteria in your sequence from the environment. And the good thing about having a speed bump is not only that your process is going to get done quicker and be cheaper, but the fact is that the process might not be possible at all without that speed up, since some processes are so large. So there's a few things about Rust that bioinformaticians might not like quite so much, so it can be a bit difficult to get going, and obviously there's a lot less online help than for Python. And some of those people are still in the lab, so they're going to be trying to do things quickly they don't really want to be spending a lot of time learning. There's less support for plotting data and stats compared to R, and less support for machine learning and deep learning, which is going to be absolutely massive in biology. And there's this kind of big perceived learning curve for Rust. But taking it all together, I consider that Rust must be really good for the world, and it's going to help our eco-friendliness as well as our economical purses. And a lot of biology is geared towards the One Health approach, which is put forward by the World Health Organization. And the idea is to sustainably balance and optimize the health of people, animals, and whole ecosystems. And by linking humans, animals, and the environment, the One Health is going to address the whole spectrum of disease control and contribute to global health security. And everyone needs to work together across all sectors and disciplines. And also, we need to make sure that we don't marginalize any types of society, because only when everything is coordinated can we properly protect health, address health challenges, infectious diseases, antimicrobial resistance, and food safety. And this will help us promote the health and integrity of whole ecosystems. So, to summarize, we talked about the potential for Rust to drive innovation and efficiency in bioinformatics. and lead us to more sustainable computing with bioinformatics in biology. And bioinformatics has got a really long way to go. If you think about the future, we might be all sequencing ourselves and handing our sequence over for personalized medicine. So thank you for listening, and I'll be on the Discord for the Q&A, and you can find me on a lot of different social media platforms. Thank you.